Thank you. Thank you to Guy for inviting me. It's a great pleasure to be here. Some friends I recognise, many that I don't. And my sort of angle or way into the microglial world is a little bit different. So I'm a neurologist. I deal with living humans, whole humans, and most of what we'll be talking about is brain imaging, some post-mortem work, and a little bit of blood work as well. And I've got no background, conflicts of interest relevant to any of the material today. But I thought I'd just give a little introduction to the diseases, because they may not be so familiar. We've heard a bit about alpha-synuclein, heard about Alzheimer's and amyloid beta and amyloid B. FTD and PSP, frontotemporal dementia and PSP, belong in a wide family of illnesses caused by pathologies of frontotemporal lobar degeneration. And they are characterized either by tau, misfolded phosphorylated tau, or ubiquitinated proteins, of which there are several, TDP43 being the most common, fuzz and others there, or 3 and 4-p tau. So a variety of different sort of molecular mechanisms. And if we think about those syndromes, what patients present with, they also have a variety of different diseases, clusters of symptoms and signs: frontotemporal dementia, progressive supranuclear palsy, corticobasal syndrome, and even these we can subdivide into different types, language and behavioral versions. Motor neuron disease comes into the fray. And in fact, we can further subdivide, particularly frontotemporal dementia, into what, from the patient's perspectives, are very different illnesses. Okay? So we've got a whole cluster of illnesses and a whole cluster of molecular pathologies. And wouldn't it be nice if they all lined up? Okay? And of course, anyone who's been in this field for a while knows it's not. It's an absolute mess. Okay? So the clinical pathological correlations are dire, very difficult. So you have this fantastic sort of genetic and molecular precision of the pathology, which simply does not map onto the syndromes. And that's a real challenge if your target for treatment were genetically mediated or had this molecular sort of protein target. The good news is that if you're approaching this from a microglial perspective, this becomes relevant across the whole spectrum. There are important similarities, perhaps some differences, but important similarities across the board. So we can actually then start to think collectively, what we would term transdiagnostically, bringing patients in across the board. And the backdrop to a lot of our work is in identifying population representative samples. So trying to winkle out everybody who's got the illness, not just those who are well enough off or well enough to come into clinics, but all those with the illness. And I think if there's time, I'm just going to show a short video of what this illness looks like, which I hope will start. How do I come across onto the... Oh, there's a delay on the mouse, which makes life interesting. Here we come.
Don't laugh. I'm not laughing.
And this is about 18 months later. It's almost mute, you hardly hear him. Wheelchair bound, tube fed.
Voice speaking? Was it swallowing?
Yeah, for him it was swallowing. So you just see how rapid and how aggressive that is. Really complex disease. You've got somatic symptoms, swallowing problems, for which he died with a pneumonia, Parkinsonism, immobility using a wheelchair, profound cognitive change and personality change with that disinhibited, almost manic appearance initially. So these are very aggressive and challenging illnesses. Our approach is to try and link together a range of different lines of attack on the illness, working with blood, and we'll actually have a slide on the cytokines at the end. I'm going to focus much of this talk on the PET imaging because it's a way in to understand the role of microglia in this illness, particularly with TSPO PET. We look at the physiology and brain structure and chemistry. I'll touch on the brain banking again at the end of the talk, very important angle. And all of this is around trying to optimize and develop clinical trials to prevent or at least to treat these illnesses. So if we just touch on some of the background, we're not going into this completely cold. There's some background indicators for immune dysregulation. I would just like to say, for the students in the audience, you can just email me. I'll send you a copy of all my slides. So you don't have to kind of be busily taking detailed notes. I'm happy to send you a copy of all the slides. But there's lots of background information from the genetic studies, preclinical models where there's information. So the P301 mutations, of which there are the equivalents in humans, have a lot of inflammation in that tauopathy. The direct neuropathology observations, and we'll see a little bit of that later. By analogy with the other diseases that have inflammation, and we've heard a bit about those already, with alpha-synuclein Parkinson's, Alzheimer's, and some early brain imaging studies. So there was lots of background information to sort of point towards an inflammatory signature, and perhaps microglia in particular. And we could draw a similar list for frontotemporal dementia. In fact, it's a very similar list. Different studies, slightly different genes, slightly different molecules implicated in the pathology, but actually all highlighting a key role of microglia. So we're going to test some hypotheses. Well, first of all, is there increased inflammation in these disorders and other dementias? Is it clinically relevant? It's another important question. It doesn't just have to be there, but is it related to how severe the illness is? Does it affect cognition? Does it affect your prognosis, your survival? And we can then ask whether it relates to other aspects of the pathology. For example, how much tau, how many tangles you have. And when does it occur? How early in your disease does it occur? We know from other background work that these diseases start to be on the move in your body, in your brain, 10, 20, maybe 30 years before your first symptoms. So how early is inflammation? How early could you go in with an immunotherapeutic strategy to try and delay or prevent the illness? So when does it occur? So let's come to those in turn. A lot of then the rest of the talk is going to focus on a PET imaging, the rationale for which goes like this. As many of you have seen already, as microglia go from their resting to their active state and their pro-inflammatory state, they lose their ramification and become amoeboid. So they have a morphology change. They also have a change in their cell surface markers. We're very interested in one on the surface called P2X7R. Also because of therapeutic potential targeting this. But the majority of brain imaging through PET over the last 30 years has used the expression, an increased expression of a translocator protein on the microglia within the mitochondria, within the microglia. So you have this TSPO translocator protein, which binds to a number of molecules that we can use as radiolabeled ligands in a PET scanner. I'm going to talk about PK1195, that's all we use here. The signal to noise isn't quite as good as with some of the second generation ligands, but it's very consistent between individuals, whereas the second generation ligands, which have higher signal to noise, also have genetic polymorphisms that affect the affinity, the net result of which is for every bit you gain in signal to noise, you lose about a third of your patients because they have the genetic polymorphism, so you actually lose out, especially with rare diseases. So we're going to use PK1195, I'll probably call it PK PET for short, and I'm going to really talk about a range of studies coming out of a program called the Nimrod program, and actually all credit to Peter St. George Hislop in the third row here for his fantastic support in getting this whole study off the ground 10, 11 years ago through the Biomedical Research Centre. None of what I'm going to show you would have happened without Peter's support. It's co-investigated by John O'Brien and myself, and many of the detailed studies I'll show today are led by Mauro Malpetti, but it's a huge team effort in the lab, in the clinics, in the scanners. The study itself concerned Alzheimer's disease, frontotemporal dementia with its different subtypes, progressive supranuclear palsy, and Lewy body dementia, and I'll show a little bit of that. And what we did with all of these people is to meet them at baseline. They'd had the diagnosis quite early in the illness. We assessed their cognition, and we followed their well-being, their cognitive ability over successive years. And we related to that to what we saw at baseline. What was their MR like, brain structure, how much inflammation, this microglial PET signal. I should also highlight, I do realize that the microglial PET, as we call it, is a shorthand. There are many different microglial subtypes. That may be important, we're not sure, but we don't for humans yet have the tools to differentiate those. So I'm going to call it a very broad class of activated microglia with the PK PET. And I apologize for those who've invested their lives in differentiating microglial subtypes. I am aware of the complexity, and we would love to have more sophisticated tools, but we don't yet. It's a very blunt tool. There's also potentially an astrocytic component to the signal, I should add, but we think what we see as increase in the patients is largely microglial driven for reasons you'll see in the last slide today. So we can look at the microglial activation, we can look at how much tau with another PET ligand, how much amyloid with another PET ligand, and some other peripheral markers. And the headline figure up front is, well, in Alzheimer's disease and progressive supranuclear palsy, you get increased signal, increased microglial activation. Cortically, medial temporal lobe in Alzheimer's disease, a little bit more subcortically in PSP. But the amount you see there, it is there increase in PSP, in pallidum, in the brainstem structures, and the striatum particularly. And the amount of that midbrain and striatal signal we get correlates with their disease severity. So this is a PSP rating scale. 0 is no PSP. 10, 20 is when you tend to hit the diagnosis, come into clinic. 80, you tend to be dying at 80, and it can't go above 100 theoretically. So it's a wide range, and it correlates with how much activity you have in inflammation. But it varies a lot from person to person. So if we follow people over time, we have some people, like the man in the video, who, when we look at his clinical severity on the PSP rating scale, accelerate very rapidly, and other patients who sort of bumble along, ill, but stable-ish, coping over many years. So what's the difference between these individuals? Well, no surprise, we've got fast or slow progressing symptoms. Let me come to this side. I've shown you that the severity depends on the microglial activation, but also the rate of change. So this middle plot is the rate of progression. It's the number of points per year you progress on your clinical severity scale. Okay? So your baseline PK PET, your baseline microglial activation, predicts how fast you will subsequently progress. Okay? And with some post-mortem work, which I'll again, time and time again, we keep coming back, trying to get some post-mortem validation that it's the right areas. There's a correspondence between the inflammatory response in the PET and the post-mortem inflammation. The very first studies we use HLA-DR. We now use IBA1 and CD68. We also showed that the amount of inflammation you have at baseline correlates with the amount of the molecular tau pathology that you have. So we can compare the binding of this tau ligand, or tau PET ligand. It's not only for the PSP tau, but I'm going to call it PSP tau for short. The molecular pathology correlates region by region with the microglial activation. So each of these dots is one of around 80 brain regions, so we can correlate region by region, either at a group level or in individual subjects with PSP, and in all cases we see a positive relationship. So areas of the brain with more tau are the areas with more inflammation. So just to sort of bring that together in a whistle-stop tour, we've seen that, yes, in PSP, there is more inflammation, more microglial activation, particularly in this classic PSP called Richardson syndrome. It correlates with severity, it predicts your subsequent rate of progression, and it correlates with this marker of the molecular pathology of tau, particularly outside the basal ganglia where there's some false positive uptake. So let's see if we can do the same sort of structure and same thinking with frontotemporal dementia. And the answer, not surprisingly, is yes. But remember I said there were 3 types of frontotemporal dementia, a behavioral variant, a semantic variant, and a non-fluent variant. They have different topologies of disease across the brain. Some are more frontal, some are more temporal. But when we use the PK PET imaging, we see increased signal in a way that corresponds to the burden of disease. So semantic variant on the right here, your temporal pole, particularly on the left, but bilaterally, temporal pole has very intense atrophy. You lose a lot of your knowledge and word and understanding corresponding to the functions of that bit of the brain. And with that loss, you get very intense uptake of the PET ligand. In the behavioral variant, which is more around impulsivity and personality change and being dysexecutive, the frontal lobe has a greater burden of pathology, and we correspondingly see the frontal lobe, rather than the temporal lobe, has increased uptake. And in non-fluent aphase, which is probably the slowest moving of these illnesses in many ways, it's a much more subtle disease in terms of atrophy, but where you do get changes is perisylving. You see a little yellow blush here? That's around the front of the sylvian fissure. It's around Broca's area, for those who've done any neurology or cognitive psychology training. Broca's area, speech production in a non-fluent agrammatic aphasia. So the location of the inflammation corresponds to the syndrome that you get. And also, when we look at post-mortem in the same subjects who come into our brain bank 2 or 3 years after PET, if we look at an area like the visual cortex that has very little pathology, no symptoms from visual symptoms, whether we have patients with a TDP43 type A or C or a tau disorder, we see neither the TDP or tau markers, nor do we see CD68 positivity. But if we move to the temporal cortex, which is a sort of core area of frontotemporal dementia, we see again an increase in both TDP43 and CD68 positivity. And we see that in both the TDP43 or indeed the 88 tau staining when it's a tauopathy. So the molecular pathology and the microglial activation go hand in hand at post-mortem as well as in the imaging. Indeed, when we do look at the morphologies, we see this is particularly the case for the amoeboid microglia, and it's not the case for the resting or ramified microglia. It's only there for the amoeboid microglia. So, neuroinflammation is associated with that burden of the tau or TDP molecular pathology. Let's come back and think about this co-localization again. If we look at I showed a sort of summary in PSP before, and there's concerns around the ligand in PSP, its affinity, but less so in these disorders. If we look at the molecular signal from the AV1451 PET ligand, and we look at that in relation to the inflammatory signal on the PK1195, we see in each of these three variants a very strong positive correlation. So although it's different parts of the brain, it's always the case that the more molecular pathology, the more microglial activation, okay? Even though the exact geography or topology of the diseases is very different. So in all these cases, we see that relationship. And in fact, if we use the distribution of microglial activation in a machine learning simple classifier, the distribution of inflammation gives very high diagnostic accuracy. It's very clear to tell who's a patient and who's a control. I mean, you can tell anyway if they walk in the door, but I mean, I'm not claiming this is a way to diagnose the disease. But the point is, it corresponds very closely that there is very clear separation. There's no overlap with normality here, okay? There's absolute clear blue water between the pattern of inflammation and your status of health. And it's there for all different types of frontotemporal dementia. Frontotemporal dementia has another advantage. Around 20% is genetic, monogenic, maybe a little bit more depending how you define a family tree, but around 20% we would have a good chance of knowing the gene. Number of genes, we've got C9orf72 is commonest, the microtubule-associated protein tau, and granulin genes, so different genes. And what we see is this increase in the microglial PET is there on all three genes. So it's not gene-specific, and that tells us it's not specific to the molecular pathology of tau or TDP43. It spans that full spectrum of disease. That's really powerful if your therapeutic strategy is going to be immunotherapeutic rather than at a molecular or gene target. So it's there in all gene types. And genes have this other extraordinary opportunity. Because we know who's carrying a gene, we can go to their kids or their siblings who are well and say, well, we know you are going to get this illness unless something's discovered too soon. You are, very highly penetrant genes. But let's see when this inflammation occurs. So that this is one published example, we have others now, that we can look in people who are 5 to 10 years off their symptoms. They are well, if you met them, you would not know, but they're destined for the dementia. If we look at say a person here who's got a tau mutation, and we use the got a 10+16 mutation, this person, and we look at the degree either of tau pathology, AV1451 tau pathology, or the microglial activation. So here's an individual in the family who is symptomatic, who's a patient, okay? And they've got very high tau in multiple regions. This is 3 different regions of the temporal lobe, but you could pick elsewhere, 3 different regions of the temporal lobe, and in each of those 3 regions you've got increased microglial activation. Okay? I've sort of shown you that on the previous slides. But here's the thing. Take a member of their family, same mutation, who 10 years before we reckon they'll get symptoms. With the tau mutation, there's quite a good correlation within family about when your symptoms start. So we reckon he's about 10 years off. There's no discernible PET detection of that tau burden. Now, the core molecular sort of biology has to be tau because it's a tau mutation, but he hasn't got an accumulation of tau aggregates and those tangles that we can detect with PET. So 10 years pre-symptoms, hasn't built up that tau burden. But look, already there is extensive activation, as much inflammation activation of microglia. So even 10 years before symptoms, at a stage when the tau pathology hasn't really consolidated, we see extensive microglial activation, giving a green light to really want to think about early pre-symptomatic immunotherapeutic strategies. So as we summarize for PSP, we get the same features in frontotemporal dementia. It's increased in all those variants. It's pre-symptomatic. It supports group differentiation. There's co-localization with the other aspects of molecular pathology. And although I focused on DLB and PSP and frontotemporal dementia, DLB, dementia with Lewy bodies, alpha-synucleinopathy, and Alzheimer's have similar issues. When we look at Alzheimer's disease and we follow our patients over the years, what we see is that the baseline PET signal for inflammation predicts how fast you're going to decline. How many points per year will you subsequently decline? This is Alzheimer's disease. I'll show you something similar for frontotemporal dementia. So it's a transdiagnostic risk. Now, I'm going to just shut your eyes for a moment. Because of time, I want to skip over the next few images. So just take 30 seconds, just as I whiz through these images, because it's a complex study. What I want to come to is the highlights. So what we did in this study, we had the PK PET for microglial activation. We used fMRI to understand brain connectivity, particularly the connectivity of the medial temporal lobe, an area of prime concern in Alzheimer's disease, and we followed people over time. And I won't go into statistical modeling, but the take-home message I find very interesting, which is the more the patient expressed a pattern of connectivity that we linked to the microglial activation, the more they had that disconnectivity pattern, the worse their cognition. Okay. So in other words, when you turn this around, it's a form of mediation analysis for those that use statistical models. It's a form of mediation analysis. The connectivity of that medial temporal lobe is what mediates the effect of inflammation on the poor cognition. Okay? So the way in which having microglial activation in your temporal lobe affects your memory is because it damages the connectivity out or in or out of the temporal lobe. Okay. And that speaks nicely, we heard in the previous talk around the role of synapses and the critical role for connectivity and this synaptic toxicity of microglial activation. But that's perhaps for other talks over this next couple of days. We see these same relationships. Microglial activation is bad in Lewy body disease as well. So there's a very strong transdiagnostic pattern. So AD, DLB, as for PSP, as for frontotemporal dementia, well, we haven't done the interventions to prove that it is harmful. Be careful of that. But it is a very bad prognostic sign. I personally believe it is harmful from other evidence. It's the evidence here that it correlates and it predicts your bad prognosis also suggests it's harmful and is not just some epiphenomenon. So these are the sort of hypotheses we set out to test. And yes, we can find there is increased inflammation in FTD and PSP and Alzheimer's and Lewy body dementia. We've shown it's clinically relevant through those correlations with severity. It affects those cognitive networks, so I skipped over that a little bit, but I did have focused on it predicting your outcome, the rate of decline. And it correlates with other aspects of molecular pathology, and increasingly we're getting the evidence that it starts very early in your disease course, many years before symptoms. So where next? In the last few slides. Well, we need to validate. We need to really validate this in post-mortem data. So although it's always very exciting when you show these changes in living patients who are early and well, it's very one step removed with brain imaging. And things like PK PET for that translocated protein and activated microglia, it's a very blunt tool. Okay? So it has some advantages in its immediate relevance to patients with the illness or at risk of the illness, but it is a blunt tool. We want some post-mortem data. We want to say, are there things we could detect in blood that would correspond with what's happening in the brain? That would be very powerful to get some scalable markers. You could track people over time and track perhaps the effects of interventions. We're very keen to look at the relationship to synaptic loss, and I think we'll probably hear more about that over these 2 days, and think about the strategies to treatment. And that it's not wishful thinking. There are immunotherapeutic strategies as a reality. This trial came into being, and it was stopped earlier, which is a great picture. It came from the mice. It came into a clinical trial in humans, although that was stopped. In Cambridge, there's an azathioprine trial in Parkinson's disease, explicitly trying to reduce people who have a high fast trajectory to dementia in their Parkinson's disease. So it's coming to trials. We're very interested in what we can pick them in the blood. So we look in patients. This is preliminary data from our first 400 patients looking at cytokines. And cytokines, you measure about 40 cytokines, but there's a sort of collective signal which we identify with principal components analysis because they change as a family. And if you look at it as a positive signal, you'll see some familiar suspects down on the left-hand side, all as part of this blood signature. And we see this is increased in all of the dementias that we've looked at so far. Okay, so there's lots of inflammatory signals in the blood. These signals, I haven't got the slide to show you, I can tell you, these signals in the blood correspond to the disease-specific inflammation in the PET scanning of each disease. Okay. So in the AD group, the extent to which an individual shows this blood-based signature correlates with your PK PET in your medial temporal lobe. The extent to which a person with, behavioural variant frontotemporal dementia, the extent to which you get low, middle, or high expression of this blood-based cytokine profile correlates with your PET imaging signature in the frontal lobe. So that's in post-mortem validation. The blood, we're interested in post-mortem validation. This is a screenshot from morning, hot off the press, and I must thank Annelies Kragerberg. I can't see here, but is Annelies here? No. If Annelies is here tomorrow, do speak to her. That what we see in contrast to controls in a patient with PSP, that the IBA1 and the TSPO are highly correlated, a marker of TSPO, that there's co-localization. And that was important as part of the evidence that what we see in the TSPO PET is actually microglial activation. We're making a lot of assumptions in the human imaging data, so we need to kind of back those assumptions up. And here we have post-mortem data where we look at the CD8 positivity in multiple brain regions in 8 patients who'd been in our PET study in their lifetime. So we kept in contact. About half of the people in the PET study came into the brain bank, and each of these lines indicates an individual. Each of the dots is a different brain region, which are different brain regions have different impacts of disease. And in every case, we see for every patient, areas with lots of PET signal in their lifetime had lots of CD8 positivity in death. So again, I think that's not really reassuring evidence that what we see in the PET does relate to microglial activation. So there we are. Needless to say, this work's been done by lots and lots of other people. I must thank especially John O'Brien, who's chief investigator of the NIMOD study, Mara, whose work I presented a lot, and Annelies for the post-mortem work, and many, many others. And to thank you for listening. Thank you.
Thank you. Thank you. Questions? Michael.
In my clinical experience in PSP patients, those which were progressing very fast were the ones which were older at onset. Did you get a chance to evaluate whether those were the ones which had higher levels of microglial activation, or let's call it PK11195
Yes.
Labeling?
So we don't get strong age signals in the PK. That maybe our numbers are too small, around 20 per group. I mean, the effects of disease are not subtle. So we get Cohen's d effect sizes 1.5 to 2. The effects of aging might be smaller, so that we might just be missing them from power. In our cohort, we have people in their 90s with PSP, and they can live a very long time. It's quite interesting, and it's unresolved about the impact of age on survival, particularly as you've got other comorbidities as you get older.
Thank you.
Yes.
Hi, James. Lovely work. Synapse density, if you could do imaging of synapses, which we're doing, but
yes.
Where would you say that lies in the correlation, closer to the neuroinflammation or the tau loss, or somewhere in the middle, if you could predict where you think the synapse loss is closely? Is it more correlated to neuroinflammation?
So I think we would start to see, well, in PSP, we see profound synapse loss, around 40%, and it's about 4% per year, and it correlates with your clinical progression. So synaptic loss is a major player in all these diseases. It's very, very clear in PSP, but it applies to all of them, and it begins very early. You get neurons lose synapses before they lose viability and die, so it's an early signal. The oligomers of tau are synaptotoxic. They're synaptotoxic, particularly in partnership with microglial activation. So the challenge we have is PET tools don't detect the toxic oligomers. They detect these late tangle aggregates, which are possibly even not particularly toxic. So we can't just do the tau PET to the synaptic PET and expect to get the answer. We are looking at the postmortem data at synaptic density in relation to microglial activation across brain regions, and there we're using the difference stage between regions as almost a surrogate for disease progression. So if your visual cortex is affected last in a person who's come into the brain bank, I mean, they're dead, they're all at that end stage, but the visual cortex is relatively less. So if you look at this across 10, 20 brain regions, you can start to explore the relationship between inflammation and synaptic loss between regions, and that's what's ongoing.
There's a question at the top left.
Hi, thank you for the talk. I was wondering, with respect to Alzheimer's disease, I know this is relatively like on the sort of cellular side of things, but like the immediate microglial response to like amyloid plaques, particularly through TREM2, like one of the previous talks was showing, like that kind of pathway is, roughly speaking, seems beneficial, at least early on. Is that kind of, that mechanism of activation, do you expect that to produce a higher TSPO PET signal in microglia, or do you expect, or is it a different type of activation that you're kind of talking about generally?
So very early in the sort of pathogenic cascade, and it's probably easier to see in the preclinical model, so the transgenic mice, you may see a beneficial and protective effect of microglia activation, or at least some types of microglia being active. What you have to remember is by the time you even get near a clinic, even to first symptoms, you're actually very far down the illness. So if you think about BRAC staging between 0, which is no Alzheimer's, and 6, the most advanced stage, there's people in this room with 0, 1, 2, 3. Yeah, I mean, some of us may be at 3. You know, by the time you walk in my clinic with mild memory symptoms, you're probably at stage 4, maybe a late stage 3. You're actually a long way down the illness. So the understanding of early versus late in an animal model is very different to when you think about patients when you're either late or very late, essentially. So there's lots of these questions we really don't know the answer to. My take is by the time you get into your clinical manifestation, it's harmful. I'm open to the possibility that had I seen you 30 years before, there may have been a beneficial effect. But it's not how I now interpret the literature of what happens once you're symptomatic. That transition might vary from disease to disease. So there's a little bit stronger evidence that in very early stages of Lewy body disease, perhaps with REM sleep behavior disorder, you might still be in the protective phase of inflammation before it becomes harmful. But this is ongoing work.
Yeah, down here. Or you, I don't know.
Why don't I go first? Thank you, that was a great talk. Can I just ask you about the blood markers?
Yes.
So do you see a correlation between blood markers and microglia PET? Basically?
Yeah, so this is blood from around, 400 people, around 150 of whom have had the microglial PET, the PK11995 PET. And I say any criticism, this is it's just a shorthand to call it microglial PET. There's lots of caveats with it, but for the sake of argument, microglial PET. What we see is the degree to which, for each disease, has a distinct pattern of where in the brain it has the microglial activation on the PET. So, but then within each disease, we do see a correlation between that disease pattern of the PET change and the signal of this cluster of cytokines. Yes, so it is what's in the blood is correlated with what's in the brain.
And by extrapolation, do you see that clinically when patients get infections that drives their progression?
Yeah, so we'd love to know that. So these patients in the NIMOS study were quite carefully included on the basis of not having had recent infections. They also don't have any autoimmune disease, there's no Crohn's or rheumatoid, so we tried to get a kind of a minimally confounded group. With history, with hindsight, I almost wish we'd taken the opposite approach. There are ways now we could look, particularly looking at, in epigenome methylated CRP, gives you a sort of integrated signal for the degree of inflammation in recent months, a bit like an HbA1c, if people use that for diabetes. So rather than just looking at your current plasma CRP, you can look at the methylation in the epigenetics. So that might give us a handle on current versus past infection, but all this needs to be untangled. Yeah.
Thanks, James. It's a wonderful talk. Thank you. I was curious about the relationship between the functional connectivity data and the microglia PET, which I think we can probably all agree, and I do appreciate your comments on that, that it's like kind of a density measure, if anything. It doesn't tell us really anything about the phenotype of those cells. So there is some, and then there was a question about synaptic density imaging. So I mean, so we've done a bit of SV2A PETs with Oliver House in London in the context of schizophrenia, and others have done that in the context of affective disorders. And you see a nice overlap between that marker and sort of connectivity patterns. So I wondered if you were, I sort of had two questions really. One is, what do you think is really happening there? Because maybe I missed it, but do you think the microglia density is changing in response to abnormal brain activity driven by synapse loss? Or do you think it's the other way round?
So this could be the whole other talk on the relationship between microglia synaptic toxicity and tau, how that works together. So my sort of brief take is that oligomeric tau is toxic synapses, microglia take up, tau which ought to be good, and yet the microglia also phagocytose synapses. They are directly harmful to the synapses, and more importantly, when the microglia die, they're a kind of super spreader event that propagate in a very intense seeding. So any one microglia taking up tau that it ought to be beneficial is probably in the long run harmful because of their capacity for further seeding. So I would see it as the microglial activation being harmful to synapses rather than the other way around.
And so what do you think sort of might be, and you sort of alluded to the J&J compound at the end. I mean, what's the key test here? Because we wonder about this as well from the psychiatry perspective. We get very excited about synaptic pruning in a similar way, maybe. What's the key test? How can we really prove these kind of?
So what matters most for your cognition is your synaptic health. It's closely related. That's the unit of memory. And it's the unit of computation for cognition. So synaptic health is paramount. So you could either want an intervention, a drug, that makes synapses resilient to inflammation or oxidative stress. You want to have the resilience of the synapse. That's one approach. So the ultimate test would be an intervention, a drug approach to make that happen. You could take a step upstream and say, if I reduce the microglial overactivation and this toxic synergy with tau inflammation and synaptic loss, you could target the microglia to protect the synapse, to protect cognition. The ultimate test is going to be a drug study. But exactly. The background of that is to show relevance. So all these...
But a specific one, right?
Oh, yes.
Because we could think of like minocycline or something, but
whole
other discussion.
That's been where it's been tried and failed. But also, the more you can be specific, the better. And also, you want to leave systemic inflammation, systemic immunity intact.
So let me take the opportunity to ask you a question that I always get and I can't answer. So what is special about the frontal cortex and FTD that the neurons there die and that your occipital cortex is spared?
Yeah, that's $1 million question. You could have a trivial answer as well. We only call it FTD, it's because it's in the front, so it's kind of reverse causation. And if the same cells had died in the back of the brain, I'd call it PCA. So actually there's a circularity in the diagnostic process. But I think there's a more interesting answer, which is there are features of whatever it is that make frontal cortical cells, with all their properties and their positions. There are molecular signatures that make them vulnerable to some pathogenic cascades. Now, part of that story might be somatic mutations. We've shown, for example, that there is greater mitochondrial heteroplasmy in the temporal lobe and frontotemporal dementia. So there's some genetic instability, which you don't find if you do standard genomics. We're looking at trying to find evidence of somatic mutations in the brain bank, so it might be genetic. I suspect that's not a big part of it. I suspect there's more interesting things about what makes a frontal cell frontal in its journey over our lifetime and development that we just don't understand.
All right, thank you.
I don't understand. Yes,
thank you.
Please tell me if you do.
All right. With that, I want to thank I would conclude the session. I want to thank all the speakers.